Welcome to the Antigen Processing and Presentation to T lymphocytes module. In this talk, we're going to learn about the processing and presenting of peptides that are loaded on the MHC Class II molecules. We will first learn about how extracellular or endosomal antigens are processed, followed by how the peptides that are formed are then loaded on MHC Class II molecules. We will also learn about the role of different proteins like II, CLIP, and HLA-DM in the peptide loading process onto MHC Class II, as well as the role of MARCH1 on the stability of the peptide MHC Class II complex. Antigen presentation is required for the development of effector alpha-beta T lymphocytes from the naive or inactive alpha-beta T lymphocytes. Antigen presentation also aids in activation of different effector functions that is associated with the alpha-beta T lymphocytes. It should be remembered that effector functions are functions or processes that help in the destruction and clearance of pathogens or also aid in activating other types of immune cells. Peptides can also be generated from proteins that are not present in the cytoplasm of antigen-presenting cells. These peptides can be obtained from protein antigens that have been internalized by the antigen-presenting cell through endocytic mechanisms like macropinocytosis or receptor-mediated endocytosis. Additionally, bacterial or viral proteins that are present in the endosomal compartments of antigen-presenting cells can also be processed to form peptides. These peptides that are formed from the non-cytosolic antigens are loaded onto the MHC Class II molecule. Now, pathogens or pathogen components can be internalized by antigen-presenting cells through endocytic mechanisms. Once these components or pathogens are taken up by the antigen-presenting cell, they can be digested when these endosomes are able to fuse with lysosomes. The peptides that are then formed through this enzymatic digestion are loaded onto the MHC Class II molecules. Not only are extracellular pathogens or pathogen components internalized and digested by antigen-presenting cells, damaged cell organelles as well as cytosolic proteins can also be delivered to their lysosome in the antigen-presenting cells. The process of autophagy involves the formation of a phagophore, that is a membranous structure that will form around the damaged organelles to ultimately give rise to an autophagosome. This autophagosome is a vesicle-like structure which will contain the damaged organelles or cytosolic proteins that are present in a cell. The autophagosome then fuses with the lysosome to form an autolysosome. The lysosomal contents will then act upon these damaged organelles and other cytosolic proteins that are present in that autolysosome and degrade them to form all kinds of different molecules, including peptides. And thus, this way, we are able to form simpler components like amino acids, peptides, fatty acids, so on and so forth, from the damaged organelles or cytosolic proteins through the process of autophagy. Now, the peptides that are generated through this process can be loaded onto the MHC Class II molecules, and they serve as self-peptides, which are able to teach the lymphocytes on what are self-antigens and what are not self-antigens. Peptides that are to be loaded on MHC Class II molecules, whether they arise from the extracellular environment or through the actions of autophagy, are present in endocytic vesicles in the antigen-presenting cells. On the other hand, the MHC Class II molecule is synthesized into the endoplasmic reticulum. Thus, the MHC Class II molecule has to leave the endoplasmic reticulum and go to endocytic vesicles in order to encounter these peptides and bind to them. It is important to note that when the MHC Class II is present in the endoplasmic reticulum, there are other types of peptides that are present there, and hence the premature binding of these peptides has to be prevented onto the MHC Class II molecule. This is done through the actions of a protein called as the MHC Class II associated invariant chain that is also called as II. II is a membrane protein, and that is what we can see in the figure in the black box, where this membrane protein is able to bind to the peptide binding groove of the MHC Class II molecules. Once it binds, it is cleaved to form a peptide, and this peptide that is formed is called as the clip peptide. Thus, because of the presence of the clip peptide in the peptide binding groove of the MHC Class II molecules in the ER, we won't have premature binding of other peptides to the MHC Class II molecules. Thus, the MHC Class II that is present in the endoplasmic reticulum is able to associate with II, which does not allow premature binding of peptides to these MHC Class II molecules, and on eventual cleavage of II, the clip peptide will be bound to the MHC Class II. Now, as it moves from the ER to different endosomes, ultimately the MHC Class II will be present in a compartment where the peptides that will be loaded onto MHC Class II will also be present. In such scenarios, the clip peptide must dissociate from the MHC Class II molecule so that the other peptides can be loaded. The dissociation of the clip peptide is promoted by a protein called HLA-DM. The association of HLA-DM, which is a membrane protein, with the MHC Class II molecule causes a conformational change in the MHC Class II molecule, which promotes the dissociation of the clip peptide. It should be noted that peptides that bind to MHC Class II must be able to bind to the peptide binding groove with a high affinity. HLA-DM is able to promote the detachment of any loosely bound peptides to the MHC Class II molecules. Thus, not only does HLA-DM promote the detachment of the clip peptide, it also does the process of peptide editing, where it promotes the detachment of other loosely bound peptides that are bound to the MHC Class II. Thus, only high affinity peptides will not dissociate from the MHC Class II and remain in the peptide binding groove of the MHC Class II. Such high affinity peptides that are bound to the MHC Class II will ultimately be trafficked to the surface of the antigen presenting cell. With this, we come to the end of this talk, where we learned about how extracellular and endosomal antigens are processed. We also learned about how peptides are loaded on MHC Class II and looked at the role of different proteins that include II, CLIP, and HLA-DM in the process of peptide loading onto MHC Class II. We also learned about the role of MARCH1 on the peptide MHC Class II stability in antigen-presenting cells.